Hello, class. Today, we will be having our virtual activity on the conventional PCR. You will learn here the basic techniques as well as the procedure on how PCR is done in the molecular laboratory. Now, first thing to do is to prepare all the PCR contents to its ideal concentration. Usually, manufacturers already have a suggestion on the concentration of each content, so you will need to dilute some to achieve the right concentration. So once you're done with that, it is best to make a table and list the breakdown of all the contents accordingly. In this activity, we are making a 50 UL reaction volume. Make sure to include in your procedure the controls. We have the negative control, the positive control, and the no template control. Take note, in every molecular procedures, always perform your samples in replicates. So in this activity, we performed our samples in duplicates. After preparing everything, you may now start. Gather all the prepared reagents on their ideal concentration and start pipetting following the prepared table. In this step, it is critical that you should work on ice to prevent the activation of the enzyme which is in your master mix. Gently pipette all the contents in a labeled sterile PCR tube accordingly. After, a quick spin makes all the dispensed contents settle at the bottom, ensuring the complete reaction during PCR. So once all the reaction tubes are properly mixed, you can now set up the thermal cycler. Check the necessary program and input the appropriate temperature and time of each PCR step, denaturation, annealing, and extension. Include as well the right temperature of pre-denaturation, final extension, and the holding temperature. And lastly, the number of cycles. So after that, you can now load the reaction tubes that you have prepared in the thermal cycler and then start running. So once everything is secured, you can now press the start run. So after the complete running, gently open the thermocycler and take out the tubes. Then, proceed with agarose gel electrophoresis to visualize your amplicons, which is your PCR product. Make 1% agarose gel by mixing 0.5 grams of agarose powder and 50 ml of the buffer. Then, microwave to dissolve the powder. Do this in intervals to avoid boiling. Once clear, that means your powder has completely dissolved. At this step, you can now add your gel red. And once your agarose solution has completely cooled off, you can gently pour it onto your prepared casting tray. Avoid bubbles in this step, and then wait for it to harden at room temperature. Once the gel has hardened, assemble it with the tank and pour the buffer on it until the maximum level. Then, you may now load your samples onto the gel. You may start with the DNA ladder. Then, continue with your samples and controls. Mix it with your loading dyes before loading it onto the gel. After you have loaded all the samples, controls, and the ladder, prepare the power supply and set up the voltage and its time. Then press run. After the electrophoresis is done, you can slowly take out the cover and then remove the gel out from the tank. Then, set up your imager and then place your gel. You may now then view your gel under the UV light and analyze your results. And that ends our virtual activity. Thank you for watching.